Hey everybody, today we're going to be talking about nucleic acids, nucleic acid structure, and DNA replication. Your genome is all of the DNA in a cell. This includes the chromosomal DNA that's in the nucleus, as well as the DNA found in other places in a cell, like the mitochondria or the chloroplasts, and in the case of bacteria, the plasmids. The genome is what's going to encode the proteome. The proteome is all of the proteins in your cell at a given time. Nucleic acid is one of the four basic macromolecules in the cell. If you remember back to the first week of class, we learned that a nucleic acid is a polymer. The monomer of a nucleic acid is the nucleotide. The nucleotide consists of a base, a sugar, and a phosphate group. The sugar in DNA is deoxyribose. It is shown on the right. Each bend in the line signifies a carbon. The lines between are meant to show covalent bonds between the carbons. If you count the carbons starting on the right, you can see that there are 5 on deoxyribose. 1, 2, 3, 4, 5. We refer to the carbons by their number names, a point we'll get back to later. RNA is also a nucleic acid, but the sugar in RNA is just a little bit different. It is ribose instead of deoxyribose. As the name would suggest, deoxyribose is missing an oxygen on its number 2 carbon. Also notice that deoxyribose has an OH group on its number 3 carbon. Nucleotides also have a phosphate attached to the 5' carbon. Once several nucleotides are added together, the sugar and phosphates will make up the backbone of the polymer. They also have a nitrogenous base attached to them. The base can be one of 4 possibilities: cytosine, thymine, guanine, and adenine. When you add nucleotide bases to one another, the phosphate group is attached to the number 5 carbon of its sugar, and then it attaches to the next nucleotide at the number 3 carbon of that sugar. You can see the backbone of the nucleic acid in the blue box here, made up of alternating sugars and phosphates. They are held together with something called a phosphodiester bond, a very strong type of covalent bond. If we zoom out a bit, you can also see the bases sticking out to the right of the molecule. As you look, you can see that there is a directionality to the DNA, meaning the part of the molecule that is facing the top of your screen is different than the part of the molecule facing the bottom of your screen. You can't just turn it around and have it look exactly the same. At the top of the screen, the free end of the molecule has a phosphate on it. The free end at the bottom has an OH group. We call these two ends by the carbon number of the sugar we talked about before. So the 5' end of the molecule is the one the number 5 carbon is closest to, and the 3' end of the molecule is the one that has the number 3 carbon closest to it. So far, we've only looked at half the DNA molecule. The 2 backbones of DNA are held together in the middle by hydrogen bonds between the nitrogenous bases. If you will recall, hydrogen bonds are attractions between hydrogen and either oxygen, nitrogen, or fluorine. Each nucleotide has another nucleotide it always bonds with, a soulmate that it likes to be with. Adenine always base pairs with thymine, and guanine always base pairs with cytosine. The reason for this is that both adenine and thymine have room for 2 hydrogen bonds. Guanine and cytosine both have room for 3 hydrogen bonds. In this way, the 2 sides of the DNA molecule are held together by hydrogen bonds between the nucleotides, a concept we refer to as complementary base pairing. Imagine a ladder. The rigid sides of the ladder are the sugar and phosphate backbones, and the rungs of the ladder would be the nucleotides. We've already learned that hydrogen bonds are individually weak, but when you have many of them together, we can hold together a molecule of DNA and keep it in this double-stranded helix shape we're familiar with. The other thing about the 2 sides of the DNA molecule is that they are anti-parallel. Do you remember how each end of the DNA molecule was different, and we named each end by the number carbon that was closest to that end? The strand on the left has the 5' end near the top of your screen, but the opposite strand is oriented so that the 5' end is down towards the bottom of your screen. Anti-parallel means that the two strands run in opposite directions. All of this structure and the way the molecule must turn to get all the bond angles correct ensures that DNA forms a double helix. So, how do we make more DNA? When we copy DNA, we first separate the two parent strands. Each side serves as a template to make a completely new strand of DNA. We use our base pairing rules to add the correct complementary base pair to each of the templates. A will bind to T, G will bind to C. This continues until you have two complete ladders of DNA. We call this mode of replication semi-conservative. Semi means partial, and conservative means unchanging. So semi-conservative replication means that half of the DNA comes from the parent strand, and the other half of the DNA is newly made. There are many enzymes that enable DNA replication to proceed. One of the most important is a group of enzymes called DNA polymerases. An enzyme called DNA polymerase III can add monomers of nucleotides together to create a polymer. But it has some caveats. DNA polymerase III cannot start from scratch, and it must add monomers to the 3' end of the molecule. DNA polymerase will allow the complementary nucleotide to base pair with the existing DNA. Then, DNA polymerase III will use energy to form the phosphodiester bond. It can only add nucleotides to the 3' end because that's the only place where a free OH group exists. Remember, a prokaryote is a cell with no nucleus or membrane-bound organelles. A bacteria is an example of a prokaryote. Prokaryotes have circular DNA. The place where DNA replication starts is called the origin of replication. The two strands of DNA will be pulled apart, creating a space between them. A replication fork will move away from that spot in both directions as the parental DNA is copied. One of the first enzymes to start this process is called helicase. Helicase is kind of like a zipper. It unwinds the DNA helix and it separates the two strands of DNA from each other. There will be one helicase molecule at each replication fork. Proteins called single-stranded binding proteins bind to each side of the newly separated DNA molecule, holding them apart and not allowing them to reform hydrogen bonds with each other. We want them to form hydrogen bonds with new nucleotides. The next enzyme up is primase. This is an enzyme that can add a few nucleotides of RNA to the DNA strand. It is not bound by the same rules as DNA polymerase and is therefore capable of starting a new RNA molecule from scratch. It will add RNA nucleotides in the 5' to 3' direction. Now DNA polymerase has a free 3' end it can use to add the next nucleotide to. It will start at the RNA primer and move along the DNA, following the replication fork and helicase. Since DNA polymerase can make new DNA in a continuous strand, this is called the leading strand. However, DNA polymerase III can only add to the 3' end of DNA. Remember, the structure of our DNA molecule is anti-parallel, which means that the opposite strand is oriented the other way. A new DNA strand complementary to the bottom strand here would be 5' on the left and 3' on the right. If DNA polymerase were to add new nucleotides to the 3' end of this strand, it would have to move towards the right, in the opposite direction away from the replication fork. Remember, our replication fork and helicase is moving to the left. The way to solve this problem is to make the DNA on that other strand in short fragments called Okazaki fragments. Because it is made in little bits and pieces, this strand is called the lagging strand. On the lagging strand, primase will lay down a short series of RNA base pairs. This is the RNA primer. Now there's a 3' end for DNA polymerase. DNA polymerase will use that 3' end and start adding complementary DNA nucleotides. DNA polymerase III will continue to add base pairs until it encounters a section where the DNA is already paired. For example, if it runs into another primer. Now we have a full section of complementary DNA. However, we have to get rid of these RNA primers. DNA polymerase I will remove the RNA primers and fill in the empty base pairs with DNA nucleotides. Another enzyme, DNA ligase, will help make the phosphodiester bonds and seal the gaps between the nucleotides added by DNA polymerase III and DNA polymerase I. An overview of replication is that on the leading strand, DNA polymerase III will require 1' just to get going, and then will continuously add nucleotides that are complementary to the parent strand to make the new DNA strand. On the lagging strand, DNA polymerase requires multiple primers and will add nucleotides in the opposite direction. This strand also requires DNA polymerase I to remove all those RNA primers and add DNA nucleotides, and ligase to seal the gaps and make the phosphate backbone whole. Here's a list of the enzymes involved in DNA replication. If you haven't already, stop the tape here and be sure to write down their name and function. In eukaryotes, the DNA is linear. The eukaryotic chromosomes have ends called telomeres, and each time the DNA replicates, a small amount of genetic material is lost because the primers at the very ends cannot be replaced. We've already talked about why this could potentially be a problem. In eukaryotes, chromosomes are linear pieces of chromatin. Chromatin is made up of DNA and proteins. The proteins we are talking about are called histones. The DNA wraps around the histones and is held in place. 8 different histone proteins with DNA wrapped around them are grouped together to make a little ball called a nucleosome. The nucleosome is like a bead with a string of unwound DNA going from it to the next nucleosome. The nucleosomes are twisted around and around to make a loop. The loops are gathered together to make the X-shaped chromosome you are used to seeing. The DNA during mitosis is condensed, so it doesn't get tangled up with DNA going to another cell. We call the condensed chromatin heterochromatin. During mitosis, the chromatin condenses during prophase. After mitosis is over, the DNA will relax again, and we call the relaxed DNA euchromatin, or normal chromatin. Some parts of the chromosomes, like the centromere and the telomeres, are always condensed or in the heterochromatin form. When DNA is in the heterochromatin form, it is not available, and in this form the genes can't be expressed. That's it for today. See you in class.